Our next speaker is Dr. Dave Segal, who's going to be talking to us about the progress toward artificial transcription factors and CRISPR therapies for Angelman syndrome. For those of you new here that don't know Dave, which is probably few, Dave has been part of the Angelman community since 2014, would you say? 15? And he has been really one of the research founders, I would say, for FAST. There's an amazing team that started the research at FAST called the FAST Fire Team, which Dave was a part of, that was led by Ed Weber and Scott Dindo and Ann Anderson, and they're actually all here. And it's really great to have Dave present to us, which is about a lot of the work that we funded years ago that is actually seeing its way to light, and some of the newer work that we funded more of the last couple years, and how that's impacting paternal activation for gene therapy, for gene-targeted therapies. So I'm very excited for us to get an update from Dave, and for those of you who know him well, we all welcome you back. Dave.
All right, Allison, thank you for that very kind introduction. So I'm Dave Segal from UC Davis, and I want to start out by saying that if any of you are planning to do the 6:30 run with me, and I know that many of you are, see? All right, we got one. Yeah, I'm really looking forward to that. I just ran on the beach, actually next to the beach, on the cement, so we don't have to run on the sand. I just want to get that across. But yeah, join me at 6:30. It'll be fun. We won't wear anybody out, I promise. And so what I want to tell you about today is some work that we're doing at UC Davis, have been doing for a little while. And it's really the work of a group of people there. So my lab is represented in the top panel there, but this also represents the work of lab done in Dr. Jill Silverman's lab, Dr. Roy Ben Shalom, and Dr. Kyle Fink at UC Davis. So we have a whole team of people and multiple labs where we have collaborative grants and a lot of data sharing and a lot of common interest in what we're trying to do here. Let's see. Right. And here's my disclosures. And my other disclosure is that I am going to talk about DNA and RNA in this talk. But I'm going to help you through it. So, we've seen a lot of this kind of thing today, but I just want to clarify a few things. So, in this world of genetics, we have DNA, and that's where the genes kind of reside. We like to think of that as the library. And then those genes will make RNA, and that RNA will eventually make the protein. So the functional thing is the protein. So UB3A protein is the protein part, but then there's a gene for UB3A, and then the RNA is made from that gene. But if I show you this cartoon at the bottom that you've seen a lot, so just to make it clear, this part in the middle is representing the DNA. And then there's the antisense RNA, or the UB3A antisense RNA. And then UB3A also makes its own RNA, right? And then that one will eventually go on to be a protein. And in most cells of the body, it's kind of like this situation. But for some reason in neurons, that big blue squiggly line goes all the way across and now is overlapping the RNA for UBE3A. And somehow that shuts off the UBE3A expression. So this is what happens on the paternal allele. Now, it's not a problem. This is actually the neurotypical case. It's not a problem because on mom's allele, mom's copy, UBE3A is expressed. So as long as UBE3A is expressed from the mother's copy, everything's just fine. But when that's not the case, and mom's allele is not there, then there's a problem, because then UBE3A is not made here because it's silenced, and then there's no other copy of UBE3A. So that's why we have Angelman syndrome. So probably everyone is familiar now with this antisense oligonucleotide, thanks to the great work from Scott Dindo's group and several others. And the ASO as you probably know, is targeted somewhere on that blue squiggly line, that RNA, and is designed to just chop off the little bit of the end of that RNA that is kind of going over the top of the UBE3A gene. So this allows the UBE3A gene to be turned on. So this has been a transformative thing in the history of this disease, right? So now there were 3 ASO trials that are currently going on, and this is a great way to be able to turn on UBE3A in its physiological location and whatever kind of regulation it has to do. It's not being overexpressed, it's just expressing itself at its normal level. It's fantastic. So, you know, maybe many of you or some of you are involved in those clinical trials as well. And, you know, certainly I've come to appreciate the value of ASOs, not just as the first step, but also the ability to like upregulate and down regulate the amount of ASO that has to be given, you know, to get the dosage right. But in thinking about, you know, the next steps, right? The next steps after ASOs, and I think Dr. Sparrow talked about this a little bit this morning. It might be possible to go from a kind of an ASO therapy to a gene therapy that might do something very similar. And so we were thinking that we might be able to do that in our lab in work led by my postdoc, Dr. Ulla Baitner. If we could use an AAV to deliver another CRISPR kind of protein called Cas13. So this is CRISPR-Cas13. Okay, and the CRISPR-Cas13 will do the same thing as the ASO, but the idea is that we put it in as a gene therapy and just chops off that end. And so UBE3A can come on like that. So the goal is to do what the ASO does, but to do it with just 1 injection. So we were working towards that. So what is this Cas13? So you've probably heard about CRISPR and all its different forms, and there's different, let's say, family members of the CRISPR family. And so usually the ones that people talk a lot about is in the panel D there, and that's the ones that cut the DNA. But we're working with the one in panel G, highlighted in red there, and this one is the Cas13, and it only cuts RNA. So it's great to, you know, use that one to chop off the end of that RNA. And we're going to put it in with AAV, and AAV has already been talked about today. But all I want to add to that is that there's been a lot of developments in the AAV field, and so many commonly used AAVs don't cross the blood-brain barrier, for example. There are other ways to get it physically across the blood-brain barrier, and so you know, some of those approaches are being used. But newer versions of AAV are able to cross the blood-brain barrier, and you can see that green, very bright green mouse brain there. So some of these are able to efficiently get in and get into the brain. This is in mouse, and then, you know, people are very busy trying to work on very similar kinds of modified AAVs that can get into human brain. So we got some funding from FAST to work for this, and so our preliminary work was done with FAST funding. So thank you for all that funding. But then we were able to get funding from the California Institute for Regenerative Medicine, which is a big source of gene and cell therapy funding for us in California. And I mention this because, you know, I was very happy to get this award because I'm very grateful for all the fundraising efforts that everyone here works very hard to do, and I'm very grateful for all the funding that FAST has given us. But to be honest, I don't think that any parent should have to lift a finger to get research for their kids and to get treatments for their kids. So thank you for everything that you do, but I was very happy to get state and federal money to work on this project because I just think it's very unfair in our healthcare system that we have to ask parents to do anything for their kids. Thank you. So our plan was to use this AAV, introduce the Cas13, And you don't have to pay attention to the data on the slide. You can see the happy faces, but there's a sad face. So when we tried this the first time and we were using the Angelman rats, that was also funded by FAST, we didn't actually see the activation of UB3A that we were hoping to see. And we didn't really understand that at the time, but then I had an interesting interaction with Jennifer Doudna at UC Berkeley. Jennifer Doudna, you might know, has won the Nobel Prize for CRISPR-Cas, along with Emmanuelle Charpentier. And so she was saying, why are you using Cas13? Haven't you read all these recent papers that said that Cas13 has some increased toxicity to it, and maybe it won't be able to be used as a therapy? So she had been developing a different kind of system that is another member of the CRISPR family called Cas or CRISPR CSM. So it's another type of enzyme, and she was willing to share this with us before it was published, and see if this would be useful in the treatment of Angelman syndrome. So here we are starting to work with her lab. This one actually is a multi-subunit protein, so a little bit unlike some of the other CRISPR proteins. And for that reason, we had to put it into 2 AAV particles. And I don't think that was a game stopper, but not preferred. But if it would work, that might be great. However, actually, this one didn't work so well in our hands. So when we put it into little newborn rats, in that red box, you can see that there's like a cyan bar and an orange bar. They should be different heights, but you see they're the same height. That means there was no effect. So that one didn't work. And then when we put it into the adult animals, you can also see those bars are at the same level. That means there was no effect. So we're still trying to figure that out. But another thing that came up along the way was, there was a so-called high-fidelity version of the Cas13 protein that supposedly fixed that toxicity problem that it had. And so we're like, oh, okay, well, we can go back to that protein. And so we retooled and did that, all did experiments again. But again, we didn't really see an increase in UB3A that we were hoping for. So we're still working on that, and we think that we have some ideas about how to resurrect that and make it a useful approach, especially even going to the science meeting yesterday. I had a lot of useful discussions with people about how we might improve that. But we also started thinking about maybe another approach, and this other approach was kind of based on work that Dr. Barbara Bayless mentioned when she was in my lab. So in contrast to this Cas13 approach that shuts off that long blue antisense transcript kind of near UB3A, the work that Barbara Bayless was working on was trying to make the zinc finger protein that shut off that transcript a little further down, kind of where it starts. And it would have the same effect of allowing UB3A to be turned back on. So in the work that she had done when she was in the lab, we were able to inject that into a mouse. We could see that the protein was active in the brain. We have one analysis of brain activity that was actually done by Ed Weber's lab. I know he's here today. He was such an inspiration for me, getting this project started. Now we have another person that we work with that does this kind of thing. But basically, it showed that it could measure the neuronal activity in the brains of these mice. And then with Dr. Jill Silverman at UC Davis, we were also able to show that it could rescue some of the motor features of this. So, that's what we had done before. But now the idea was we were going to deliver this with an AAV and try to make it more efficient to get into the brain. So in more recently published work, we were able to show that we could deliver this into the brain. And I'm sorry, I don't have a better slide here, but if you can see like these orange bars there, see that they're not the same height as these other bars around them, right? That's the antisense transcript, and that shows that it did drop down. We can also see in these pictures of the mouse brain, and I think Dr. Alison Barrett showed this earlier, You can see that like there's a wild type mouse brain on one side, there's the Angelman mouse brain in the middle, and then on the other side, you see this kind of brown treated brain, and that means that the UB3A was expressed in that brain. But is that expression enough to actually fix the mouse? And so we looked at that too, and we were able to see, if you look at the red line, so the red line is the treatment, and the black line is the wild type mice. So in these two assays, which were looking at motor function, we were able to see that we got pretty good restoration of motor functions in this open field test, as well as this kind of rotor rod test. So it seems like the amount of UB3A that was being reintroduced here was really having a good therapeutic benefit. And the amount that antisense was turned down, it wasn't completely turned off, it wasn't shut off, it wasn't a knockout, it was knocked down. So the antisense was still being expressed, but it allowed enough UB3A to be expressed that it looks like we're able to fix at least some of the symptoms of Angelman syndrome in mice. So in conclusion, we think we have a couple of ways to work this now. One of them might be with Cas13. We don't have that one working right now, but we think we have some ways to get that back. And the artificial transcription factor is coming back. Thank you very much.
Awesome. I think we have time for one or two questions, and then we can save the rest for the panel. Give you my mic.
What complications do you see in turning on Dad's latent gene in an individual that has UPD, two copies of the paternal 15?
Yeah, so the concern there is that if we're reactivating Dad's copy and you have two of Dad's copy, then you might have twice the amount that's there. I think that we don't really know the answer to that, but there's probably a range in which UB3A is not going to lead to autism, as in the case of DUP15Q. So, we're trying to hit within that range. And again, this is in the mouse study, so we'll have to see how this translates up. That's definitely something that we're keeping an eye on. But we think that we should be able to get it to a level that it's not causing other problems.
Hi, Lisa Schaefer, Cerebral Therapeutics, and very new to this field, so thank you for giving me the opportunity to ask this question. Curious about, well, AAV would obviously be a holy grail, a one and done type thing. What are your thoughts about sticking with the injectable protein, ATF, until we know more about the dosing and regimen, if we could dose that directly into the brain, like ICV or intrathecal?
Yeah, I think that's great. And so the work that's being done with the ASOs is already like piloting the way. And we're learning so much from what we know. I mean, I don't know a lot because I'm not associated with those trials, but what is known publicly is really lighting the way for all therapies that come after that. And so I think that those are great ideas. Yeah.
Thank you for your talk.
Thank you.